All right. Welcome everyone to today's webinar. And today I will give you a basic introduction to proximity network data analysis. And let's get started. So we have a few other webinars available online on our YouTube channel that you can look at for introduction to our latest kit, the Pixelgen Proxiome kit. Because today's seminar is completely focused on the later part of this workflow covering the data processing and the analysis. But just a quick background. So the proximity network assay that we have developed is a technique that focuses on targeting the surface proteins by having barcoded antibodies. And these antibodies are then connected with the linker sequences and generating a sequencing library that contains information both about the protein that is targeted and how they are linked together in space. And with this information, we can generate networks for every individual cell that we are processing. And with this kit, we have the Immuno 155 protein panel. And this contains 155 antibodies covering or targeting a wide variety of well-selected. They are mainly focused around the different immune functions. And for each of these proteins, we are then able to measure the protein abundance, but also the spatial information about them. And we refer to our spatial scores as either clustering when we talk about the self-proximity of a protein, and we measure the co-localization between two proteins. So for this, we get 155 times 155 matrix with information. So for today's seminar, the outline and the goals is for you to understand the basics of the proximity network data analysis and what available resources that we have, and then give you a few examples of proximity network data analysis that you can perform. So I'll start off by introducing the data processing step. And since the technology is sequencing based, the main output file that we get are sequencing FASTQ files. And these files are processed with our Pixelator pipeline. And this is a pipeline written as a Nextflow pipeline, meaning that it's open source and available online. And it outputs a lot of different files, but the key ones are the QC report and then the pixel file. So if we take a closer look at what the Pixelator pipeline actually does. So to summarize, it takes the FASTQ file read information and process them to map the correct proteins and how they are linked together. And from this information, it can generate and build these big networks for every sample. And in these graphs, the nodes are corresponds to the proteins, and how the proteins are connected to each other is the edges between the nodes, which are the actual linkers that we introduce in the technology itself. And then the next step is to take this big graph and split it up into individual components where you have highly connected individual subgraphs. And each of these components then corresponds to single cells. Within the Pixelator pipeline, we also compute our key proximity scores. So these are the spatial metrics that we have to describe how the proteins are located in relation to each other. And here we talk about the clustering and the co-localization. And to compute this, we are actually just counting the number of times that each of the protein are connected to with one another through this linker. And by summing this up, we then get our observed join counts, as we term them. But then to get some statistics of it and understand if this is a signal that is significant or not, we compare it against a random distribution. And we generate this random distribution by shuffling the node labels of the graph and repeating that process multiple times, and then computing these join counts for the random distribution. And with those metrics, we can compute a z-score. And this z-score is our proximity score, and having a positive z-score means that we have a clustering or a co-localization that is more prevalent than you would expect by random chance. And if you have a negative z-score, that means that you have a protein or a co-localization that is more segregated or dispersed than you would expect by random chance. So on our software page, you can go into depth and read more about these algorithms. And then for the final step of the pixelator pipeline, we also compute the graph layout for every individual cell. So this is how we are able to generate these 3D representation of what the cells actually looks like. And then in addition, we also generate a lot of different metrics that are important for downstream analysis. And this includes looking at the number of the total number of molecules that we capture per cell, the sequencing reads per cell. We have some metrics to evaluate the antibody aggregation. And we also have included 3 different isotype control antibodies within the kit. And these are used to measure the background level of non-specific binding. So we can report that fraction as well for every cell. And then we also provide a metric for the graph connectivity. And this is something we refer to as the coreness or the K-core of the network. And it basically means that, or describes how well connected each of the proteins are with each other. So the higher connectivity or coreness you have of the data, the more densely connected the network is, and the more robust and reliable it is. So as I mentioned, this pipeline is a next flow pipeline, meaning that it's available through NF Core. So you can head to nfcore/pixelator to have a look at how to install the pipeline, and all the documentation of it is available online there. And this is the outline for all the steps of the pipeline. And as you can see, there are 2 different paths, one for molecular pixelation data, which is our previous technology, and now for the proximity network assay, the PNA. So that is the blue line that is of relevance for today's work. And as I mentioned, the key input is the FASTQ files. And then together with the panel file that is provided within the pipeline itself, you set up this sample sheet. And that's more or less all you need to initiate the pipeline. And from that pipeline, you get these different output files, where the main one is really the pixel file. And in addition, you get this summary or quality control report, which is an HTML file. So that leads us to our next step, which is the quality control. So the first thing that we usually look at when we have run the pipeline is this HTML file. So it's a file that you can open in your web browser and gives you a great quick look at the data and the quality of the data, as well as information about the pipeline run. It includes a lot of useful and important statistics around the quality, such as the number of reads and the number of cells that you have, sequencing saturation, and the number of unique proteins that you capture per cell, as well as the network connectivity and a lot of other important metrics. And then when we move on, we start to look into the pixel file. And as I said, this is the key file that we work with. And it's the file that contains the information about protein abundance, all the proximity scores and cell level metadata. And within the metadata in this file, we have all the important quality metrics, such as the UMI count and read counts, and also information about the isotype control levels, network connectivity, and this antibody aggregation score. So if we have a look at one of these metrics, so one key thing that we typically look at is the number of molecules, or the UMI count across all the components or the cells. So we generate these rank plots, and it allows us to set an appropriate cutoff for when to remove components that contains too few molecules, and determine which components can be regarded as normal cells. The other thing I mentioned is the isotype fraction. And in components with a very high degree of isotype antibody binding, we determine them as having too high of a non-specific binding of these antibodies, and they are not very reliable in that sense. So we typically filter based on the cutoff here, and try to keep cells that have as low isotype fraction as possible. And then we move on to the actual data analysis. So for this, you can either continue using the Pixelator library, or you can use our own package called Pixelator R. And here again, you can go to our software site, and we have a lot of tutorials for how to install these packages, and descriptions of how to use them, etc. So again, the Pixelator pipeline, or the library, which it actually is a Python library, and it relies on the ScamPy library. While if you're a R user, we have Pixelator R that we have developed internally that relies on the Seurat framework. So if you come from the single cell RNA-seq world, then these packages will probably be very familiar to you. So for the actual downstream processing and analysis, we follow a few key steps, starting with the quality control, as I mentioned, and filtering of low quality components or cells. Then we do account normalization and scaling, and here we typically recommend users to use the CLR or the DSB approach. And then you can proceed to do different kinds of dimensionality reduction, for instance, a PCA followed by UMAP to project the cells in 2 dimensions. And also we typically recommend to do different types of batch correction or data integration to align the different samples if you have multiple samples that you're working with. And then you can do different types of cell clustering, or defining your cell populations using the abundance data or gating based on known cell markers. And finally is the actual spatial analysis, which is the unique aspect, of course, of our technology. So if we take a closer look at these 3 components, the abundance, clustering, and the colocalization data. For the abundance, that's the more traditional way of viewing single cells. So here we just look at the levels of different proteins within our cells, and we can use this to categorize or group the data into known cell populations. And by having this 55 marker panel, we are able to get a very high granularity of the cell clustering. And then, of course, you can look into the abundance of all these different proteins and in the different cell populations. And you can do differential comparisons between populations and between conditions if you're interested in that. And then moving on to the more unique aspects of our data is to look at the proximity scores. So starting with the clustering, here we have an example of a data set that we have generated in-house, looking at T cells that are either in resting state or been activated. And here we found the CD82 protein to be of particular interest. So if we look at the abundance level, which is what you typically look at with other techniques, it remains more or less unchanged, depending on if it's resting or activated. But when we instead look at the clustering score, here we see an increase in the CD82 clustering in the activated T cells. So we could also look at this spatially on a single cell level, and we could see that CD82 forms these aggregates. And this could also be confirmed by microscopy. So this is an example of where this technology provides novel insights that would otherwise probably not have been studied in this way. And yeah, then we move on to the co-localization. So here we can look at all 155 markers and how they are related to each other. So in this heat map, every dot that you see here corresponds to the co-localization score, or the Z score, between two proteins. And then in the diagonal that you have here, that refers to the protein clustering. So when you have of the same protein identity. And when you visualize it as these heat maps, you can also do hierarchical clustering of the data and start to group the proteins into domains where you have or shared co-localization scores. So to be able to identify then proteins that co-localizes strongly with each other and forms these different multi-protein domains. Another way that we can compare the proximity data is to do differential analysis. And here, for instance, we are comparing the activated versus the resting cells. So having, on the right side, we see then proteins that co-localizes more strongly in the activated cells as compared to resting. So here you can start to appreciate differences in your data and the changes that occurs in the organization of proteins. And we can also visualize this as different types of networks. So here we generate a network based on the proximity data itself. And we can compare how this changes then in the two different settings. So this is one way to look at the co-localization patterns. But of course, the possibilities for doing downstream analysis is very broad. So another example that you can do is to, instead of doing cell clustering based on protein abundance, is to do a proximity-driven cell clustering. Or you can start to move towards doing multimodal data integration. And finally, I wanted to give a quick description of a case study that we have. And this is part of our application notes. So if you're interested in this particular case study, I would highly recommend the webinar by my colleague that was given recently, where she goes over this study in depth. And you can also find the application note online, and it's gonna be out as a preprint on bioRxiv very, very soon. So in this experiment, we took CD19 targeting CAR T cells and we co-cultured them together with cancerous Raji B cells. And this allowed us to study the interaction between CAR T cells and the tumor cells, and also to identify this phenomenon known as trogocytosis. So for the data analysis here, we could study this trogocytosis phenomenon where you have patches of the tumor cells that gets attached to the CAR T cells. So we can see that by identifying these very dense clusters of B cell markers onto the T cells. And we could also start to quantify the number of patches that we identify and see that with the co-culture, you have an increasing number of patches, and especially in the exhausted T cells. And we could also identify and visualize cell conjugates. So this is where we manage to capture two cells that are still attached to each other. And you can see how one end of the network is enriched for these B cell markers, while the other end carries the T cell markers. All right, so the final step here that I also wanted to include here is the data visualizations and some examples of what you can do with the data. So I've already shown you some examples of this, and one thing that we typically generate is these types of proximity heat maps. So this is to summarize as much data as possible within one single plot, and you can start to identify these compartments of co-localizing proteins. And we can also do these differential comparisons on the proximity data and compare different cell types, for instance, or conditions, and see how different proteins are differentially clustered in this case. And then we can also, of course, generate these proximity networks to try to summarize how groups of proteins co-localize or segregates from one another. And finally, what I think is the most asked for, or what people like the most about this technology is, of course, the ability to also study our cells, or the cells that you process in 3 dimensions. So here within our R package and the Python libraries, we provide functionalities for exploring the cells in 3 dimensions interactively. And you can also generate figures for multiple cells, of course, to try to identify consistent patterns. And you can also visualize these cell conjugates. So that's a few examples of how you can visualize our data. So to summarize, the proximity network assay generates very rich and unique data type. And for you to analyze this data, we provide accessible tools to do the downstream data analysis and processing. And this is guided by very detailed tutorials and that you can find on our software site. And then at Pixelgen, we also believe strongly in open source and open data formats. So we hope that users will continue to explore new analysis and explore new algorithms for analyzing the proximity network data to identify new things and potential mechanisms for cell functions and processes. Yes, so if you're interested in learning more and have questions for us, you can always send them to us at hello@pixelgen.com. And this is the Pixelgen Proxiome kit. So if you have any questions about the kit or the data analysis, feel free to reach out to us. And that was everything for today. So thank you very much for listening, and happy to take any questions.
I see a question here in the Q&A panel. Someone has asked, do the proximity heat maps show aggregated scores of multiple cells in the data?
Yeah, so in the ones that I showed here do have aggregated. So it can be a mean proximity score or a median. You can decide yourself. It's very flexible, of course, how you choose to present your data. But you can also generate a heat map like this for every individual cell if you want to. So it's really up to you. But in this case, it was aggregated for the entire sort of cells or cell populations, yeah.
All right. I can ask a question. Do we need certain kind of bioinformatic expertise or resources that are necessary to perform this analysis?
Yeah, so we typically recommend you to have a bioinformatics person to help you out with the data analysis. I would say it's quite similar to processing and analyzing single-cell RNA-seq data. So ideally, for now, having someone that is familiar with either and comfortable with either like Python programming or R programming will help a lot, and having experience working with single cell data in any form. And then for running the pipeline, for instance, you also need access to some sort of cluster or a more powerful sort of computer to actually run the pipeline. But otherwise, it's quite straightforward, and we have a lot of tutorials online, and we're trying to make it as user-friendly as possible. Yeah.
All right. We have some more questions here. Daphne has asked, thank you. But when we say components, do we always mean cells?
Yeah, so that's a good question. So components is, of course, a very technical term, because we don't do any type of cell identification before, like within the technology itself. So we identify the cells using this graph algorithms and to extract these components. And then in some cases, the components will be very, very small, so they might correspond to only parts of the cell. So that's why we need this filtering based on the number of molecules, for instance. But then what you have, or like in most of the cases, the majority of the cases, one component will really correspond to one single cell. I also showed you these cell conjugates. So that's an example where you have one component, but it's actually comprising of two cells. And there you could apply different duplicate prediction algorithms, for instance, to also help identify these duplets. But yeah, so that's what we mean with the components and the cell sort of terminology, and how we mix those terms sometimes a bit. But the components is the more sort of technically correct term.
All right. Next question is about the proximity network data. Vaishnavi asked that, is it closer to transcriptomics or proteomics in complexity, our proximity network data?
In complexity, I'm not sure if I understand correctly the question, but I mean, it is a proteomics technology, of course. So,
Yes.
and it's yeah, I don't know if I have a good answer to this, but I would say more towards proteomics. But from my own end, I'm more used to working with RNA-seq data and transcriptomics. So yeah, I can't say for sure, but
Was
there
it
is a lot of
difficult?
similar. Sorry.
I was going to say, was it difficult? Is it more difficult to work with transcriptomics data, or is it more difficult to work with our kind of data?
Yeah, I mean, in terms of complexity of working with
Yes.
it, and
Yes.
yeah, okay, yeah. So no, then I would say it's quite similar. Like the way it's designed and this technology works, it's very similar to the streamline or the type of analysis that you do for single cell RNA-seq or transcriptomics. Since it is a sequencing based technology. So if you compare to other like proteomics methods that is maybe based on mass spec or imaging or something else, I mean, that's another type of complexity to those analysis often. So in that regard, yeah, absolutely. Then it's more like a traditional kind of sequencing based transcriptomic method. Yeah.
I don't see any more questions, but I have one last question. So do you experience a lot of issues stemming from batch effects, and how do we resolve these?
Yeah, so I mean, it comes back to your experimental design as well, but in most biological experiments, you have differences that you see between samples. So that's why we typically apply algorithms for doing batch corrections and sample integration. But we mostly use, I think I mentioned in the slide, this harmony package, which is very well integrated in, for instance, Seurat and the workflows there. So that takes care of most of the batch effects. We don't see a huge amount of batch effect if you run all the samples together from one kit and then sequence everything together, then there is very little technical batch effects. But of course, you will have biological differences if you have samples from different patients and different cell types and so on. But here you can really turn to a lot of different algorithms that are available for doing batch integration. So there's been a lot of tools developed for single cell transcriptomics that you can try out as well to do this data integration.
Okay, we have one more question here. So when Z-score is calculated, is it calculated for each component separately, or it determines clustering of components?
It's for per component. So you get this score for every cell or component in your data set. So this is a summarizing metric, and the good thing about it is that it really, I mean, it balances out or takes into account the abundance of your protein, so it kind of normalizes against biases in protein abundances, and also then given the architecture of the network that you have within the cell. But yeah, it needs to be computed on a per cell or per component level.
All right. Vaishnavi, thanks. I think she got her answer about the complexity question. She says that she also has an experience with transcriptomic data, which also uses linkers and it has a sequence-based approach. All right. I think we have answered all the questions that came in. And thank you everyone for joining. And if you still have questions, please send us an email on hello@pixelgen.com. We are very open to answer all your questions. I'll add this recording to the website as well, so you can revisit it and listen at your own pace. And thanks for joining today. And if you need a web seminar at your institute, if you're interested in knowing more about the technology, please send us an email as well. We can organize something. Thank you so much, Lovisa.
Thank you all.
Okay, bye.